Good afternoon. My name is Meghna Stepanian. I'm the President, CEO, and founder of PROMETRIKA. Welcome to the first of our 3 webinar series on natural history studies for drug development. Just a word about PROMETRIKA. We are a full-service clinical research organization located in Cambridge, Massachusetts. And for the past 18 years, we have collaborated with our sponsor companies in developing hundreds of drugs and biologics. I'm joined today by my colleagues and our guest, Dr. Mark Hurt. Mark is a consultant specializing in drug development in neurological and rare diseases and the former CMO of Alceris. My colleagues this afternoon joining me are Nicole Lavallee. Nicole's the Senior Director of Biostatistics at PROMETRIKA, and Chelsea Ryan. Chelsea is the Associate Director of Clinical Research and Pharmacovigilance at PROMETRIKA. Just to give you an overview of the next 45 minutes, we will look at the ways that natural history data is used in drug development, look at the different types of natural history studies, touch upon operational challenges and lessons in operational conduct of natural history studies. And we will devote the majority of our time to the case study that we are featuring today, highlighting the unexpected challenges of designing natural history studies during COVID-19. We will touch upon how we were able to leverage a successful industry sponsor collaboration to develop a creative solution for these challenges. So what are the uses of natural history data in drug development? From my perspective, having done this for many, many years, the 3 key areas are to first and foremost gain information to help design, usually the first randomized controlled trial in a drug development program. And specifically to select the appropriate efficacy endpoints and to come up with the timing of the assessments. Also, to get an idea of the variability of those endpoints and to aid a successful and meaningful sample size estimation for your first randomized clinical trial. Another very important use of natural history data can be to establish a surrogate endpoint for accelerated approval. That usage really focuses on establishing how the change in a biomarker that's being considered as a surrogate endpoint can predict a change in a established clinical endpoint. And usually these types of analyses involve more than one natural history study. They involve multiple studies that are pooled using meta-analytic techniques. And finally, a use of natural history data in drug development that is of great interest to many of our sponsors is to use the data as a historical control arm for a single arm clinical trial. Here, the key is matching of the inclusion-exclusion criteria between your single-arm clinical trial and the natural history study that you are utilizing, essentially to step in for a placebo arm that is challenging to have in your first trial or in your pivotal trial even. And propensity score-based methods, statistical methods are used to mitigate the fact that there is no randomization. So what are the types of natural history studies that are commonly done in our industry? Many sponsors start with a prospective natural history study where while the study is non-interventional, there is no study drug involved, but pretty much other methods used in the conduct of the study mimic and are similar to the methods that we used for clinical trials. Another type of natural history study is a disease registry where the methods used are observational in nature. Both prospective natural history studies and disease registries are prospective. They are planned, and after the point of planning, data are collected. But another option is to leverage patients' records, basically medical records, and to conduct a retrospective natural history study. And that's another type of study that still requires a protocol and formality, but once the study is planned, you are looking back and not forward. These studies have commonalities and differences. These different types of natural history studies, and they have commonalities and differences with the clinical trials that we are all familiar with. So just a comparison of the key characteristics of randomized clinical trials versus natural history studies versus single-arm trials that use natural history studies as their historical control arm. Well, randomized clinical trials we all are familiar with. They are interventional, randomized, and prospective. Single-arm trials are interventional. They're, of course, not randomized. They are prospectively designed. And they employ more natural history data than, let's say, just a randomized trial that has 2 or more arms being compared. That still collects some natural history data in form of medical history. And then prospective natural history studies are not interventional, not randomized, but of course they're prospective. And everything that they collect is natural history data, but they're collected moving forward. Registries are very similar. Again, just the methodologies used are slightly different in terms of their rigidity per se. And finally, retrospective natural history studies are not interventional, they're not randomized, and they're not prospective, and they, collect a great deal of, shall we say, free-form data looking back into the records of individual patients. Now I will pass this along to my colleague, Chelsea Ryan, to talk about the operational lessons of natural history studies, specifically more focusing on prospective natural history studies. Chelsea?
Thank you so much, Meganoush. Now that we understand the types and design and data collection approaches of natural history studies, I'm going to provide a quick overview of our operational lessons learned from our clinical operations team with collectively over 30 years of experience working on these types of studies. In our next webinar, we will drill down a bit more into the operational challenges. And if you have any specific challenges that you would like us to cover, please feel free to send those to us to consider as well. In our coming slides, Nicole and Mark will be reviewing a case study of prospective natural history study, as Meganush mentioned. In our experience, common pitfalls of prospective natural history studies fall into one of these four categories: patients and PIs losing interest, overlooking regulations, failure to consider real-world behaviors, and undervaluing technology and the use of study vendors. The underlying common mitigation strategy is to treat natural history studies with the same amount of rigor as one would a clinical trial. This is especially critical when considering future uses of the data collected during a natural history study. The study team must understand the patient population and their daily life, and a cookie-cutter clinical trial approach will not be successful for these types of projects. It is especially important to partner with stakeholders who understand the operational strategy must be fit for purpose. Our clinical operations team has extensive experience employing protocol-driven natural history risk mitigation strategies, including but not limited to retaining engagement through the sharing of study newsletters and interim results, applying risk-based monitoring and quality management methodologies, engaging with diverse groups of stakeholders, and centralized data collection through ePROs, patient diaries, and central labs. I'd like to now hand it over to Nicole and Mark to review their case study experiences. Thank you so much.
I'm Mark. Nicole and I are going to describe a case study where we talk about unexpected challenges of designing a natural history study during COVID-19. Here's our story. It has a happy beginning, a crisis in the middle that involves heroic effort to resolve it, and then a happy ending. In our story, the sponsor realizes that published natural history has intervals that do not match with our clinical trial plan. So the sponsor begins a natural history study to select variables and duration that matches that for a phase 2 study. I want to emphasize here that the drug development program that we're describing to prevent a cancer chemotherapy. And therefore, when we talk about drug exposure and cumulative dose, we're going to be talking about the cumulative dose of a toxin, not the cumulative dose of a therapy. And you'll see that every once in a while we talk about exposure to drug, we're talking about exposure to the toxin, not exposure to a therapeutic. In our case study, the sponsor has been using best practices. They are doing all of the things Chelsea has recommended to make sure that the conduct of the study goes well, that the data is collected in an organized way, and that the data is going to be interpretable at the end of the study. The sponsor is running a study, the 12-week chemotherapy regimen. They hope to get 40 subjects into the study. They plan assessments at baseline and every 2 weeks during the 12-week chemotherapy regimen. They collect clinical assessments, patient-reported outcome scale, and a biomarker. The sponsor expects to collect a very rich data set, 40 subjects at baseline and at 6 post-baseline time points. So this graphic shows you the number of subjects and the number of time points they expect to collect and analyze. The crisis comes when COVID-19 shuts down the study in March 2020. Data collection grinds to a halt. Study completion is no longer in sight, and only 10 subjects have completed the 12-week chemotherapy regimen. Now the anticipated data looks like this. a 75% reduction from their expectation. So the sponsor shifts to Plan B and decides to collaborate with an academic investigator. This academic investigator has a natural history study already in the can with an N of 30 subjects, but a small number of time points. And now the anticipated data set looks like this, where the blue diamonds are the sponsors data set and the red diamonds are the academic data set. At this point, things have become complicated. We need to involve statistics specialists because the large CRO is unable to pivot. Once experts are engaged, it's very important that we communicate on several channels by voice, bullets, mock tables, mock figures. We need to write things down and make decisions about how we're going to proceed to analyze the data.
So I enter at this point, and before writing the SAP, it's necessary for me to become familiar with the data. I'm already familiar with the sponsored data at this point, but the academic data comes to us in Excel. So it's necessary for me to look very closely at that data to become familiar with the contents, as there's no documentation accompanying the data. So I need to compare the sponsored data and the academic data to see what's in common and what's different and what's combinable. So there's a few different challenges that we faced with this academic data. First of all, the data was entered in Excel. So there was no documentation accompanying the data. And in addition, there was no data management plan, which means that there was no written standards for either the data entry or for the cleaning of the data. In fact, when we got the data from the academic site, the dates that we got were both in US and European formats. Had we not spent some time trying to get familiar with the data, we may not have noticed that. So with the sponsored data, the visit schedules were very consistent. With the academic data, the intervals between visits were very inconsistent, pretty much very different from patient to patient. So there was no analyses that were possible by visit. And then we received Excel spreadsheets rather than SAS data sets. So again, there was no labels in the data sets and we had to read those into Excel.
So based on this from the literature, we're interested in the relationship of each response variable to the cumulative dose of the toxin. And here the cumulative dose is in 6 bins, and there's a box and whisker diagram of the ClinRO shown for each box.
Now the advantage of actually doing the cumulative dose bins is that we've already seen at this point that the sponsor and the academic data cannot be combined based on visit. So the cumulative dose is actually a way of standardizing and combining the data that works out quite nicely. And then we can see based on this plot that there is a very strong relationship between the cumulative dose and the ClinRO score.
And of course, we're interested in the time course of each of the response variables. And here we show the mean biomarker and the mean ClinRO that are displayed on the same plot.
And it's very interesting here to see how closely the mean biomarker A and the mean ClinRO score actually track together. So what's important to do now is to actually put the academic data and the sponsored data side by side and kind of compare them. So we did that on a number of outcomes, but this particular outcome we're looking at now is the weeks on treatment. And what we're seeing here is that up to week 12, the data is largely from the sponsor study subjects, whereas after week 12, it's largely the academic data that we're seeing. So this is something that wasn't expected. So we're wondering, is this correct? So we have to go back and ask.
And at this point, we contact the academic investigator who advises that we speak to the data manager, who happens to be an MD. And in this case, I think MD stands for Mon Dieu.
So this is the data that, well, it's a depiction of the data that we got. It's actually a nice depiction. It's actually the visit schedules for the academic subjects are depicted in red here. They were actually much more dispersed than what's indicated in this graph.
At this point, we pivot to Plan C. We decide to split the data sets for two reasons. First, the logistical issue of correcting the dates in the academic data. And second, there's the scientific opportunity to develop hypotheses in one data set and test them in another data set.
So at this point, we've actually changed the statistical analysis plan a couple of times. Now, normally that would be a concern, but in this particular area of research, which is not very mature at this point, we're still developing hypotheses. So the regulatory authorities will be, willing to relax a little bit on the changing of the statistical analysis plan. What they won't relax on or give you leeway on is the quality of the data. So it's very important at this point that we go back to the academic site and make sure that the data that we have is correct.
We have some research questions. We want to know how the biomarker, the patient-reported outcome, and the ClinRO change over time. We want to know if we can build predictive models for each of these, and we want to figure out which outcome variable we should use in a proof of concept trial of 4 weeks or 12 weeks duration.
So how can we build statistical models to answer these questions? So the first thing we consider is what measures are in common between the sponsor and the academic data set. Both data sets have both the patient-reported outcome measures and the clinical reported outcome measures, but they only have one PRO and one ClinRO measure in common between the two. So those are the measures that we're going to include in our models. In addition, we look at some graphical analyses and correlations between some of the predictors to see what the relationships are and which have high correlations. A third factor that we need to consider are the sample sizes. We know that we have about 19 patients in the sponsored data and 27 patients in the academic data. So we need to be careful with over-parameterizing our models because with so few patients, having too many parameters in the model, you're not going to be able to fit the models correctly. So here are some of the scatter plots and correlations that we looked at, and these are produced by SAS-Proc SG Scatter, a very easy procedure for anybody to use. It produces these pairwise scatter plots as well as the pairwise correlations, and you can see from these correlations that there are a number of pretty strong correlations of 0.5 or above. So what we're planning to do, we haven't done this yet, but we're planning to do basically a split sample model validation. So while the academic data is being cleaned, we're going to build the models using the sponsored data. So we're going to use SAS proc mixed, and basically we're going to fit growth models to the data. We're going to have separate models for biomarker A, the PRO, and the ClinRO assessments. Predictors will include cumulative dose, time since first dose, and baseline covariates. And then we're going to use goodness of fit statistics to compare models and drop variables that do not add to the predictive capability of the models. Once the academic data comes back and it's clean, we're going to rerun the same models that we built using the sponsored data. And we're going to check that the model fit and the parameter estimates are consistent between the sponsor and the academic data. Now, what I want to stress here is that this model building step is actually probably the easiest and least time consuming of the process, of the entire process. Everything that came before, the data familiarization, the descriptive analyses, the data cleaning, and the data integration, all those steps were much more time consuming, and they needed to be done very carefully and thoughtfully. Otherwise, any model building that we do is basically of no value.
So again, here's the story. It boils down to this. Mark is a man with a problem. He's got a truncated data set and he needs to supplement it and then analyze the supplemented data sets. Nicole is a woman with solutions. All's well that ends well.
As we have demonstrated, to successfully execute a natural history study, the study team must have a robust, fit-for-purpose plan. This requires partnering with the right knowledgeable and experienced partners as early as possible in the process. The case study shows us that you can mitigate and plan for all of the common pitfalls: losing patient and investigator interest, lacking regulatory rigor, failure to engage a diverse set of stakeholders, and undervaluing technology. And still, if you do not collaborate with experienced partners, you will likely run into issues. One of the keys to looking for a CRO partner is to make sure you are on the same page, to not treat this as a typical clinical trial. It must be handled with experienced care and intentional strategic planning for current and long-term uses of the study data. The same data management and clinical operational plans for a typical clinical trial do not work for a natural history study, especially for rare disease indications. In our next webinar series, we will dive deeper into discovering ways to think outside of the box when designing fit-for-purpose natural history study plans. Please stick around for our Q&A following this session. We will also be sending a follow-up email to this session with time and days each of our speakers will be available for you to set up a quick call if this is something that you're interested in as well. Thank you so much.
Thank you so much, Mark, Nicole, and Chelsea. It was a very informative discussion and very informative presentation. We will now go to our question and answer session. And I will start it with a question from myself to Mark, but I encourage our audience to keep sending us your questions and we will answer them in the order that they arrive. So Mark, you have been a CMO in pharmaceutical companies. You have been a leader in drug development, and many of these programs that you've worked on have started with a natural history study. Can you tell us, how do you just kick this process off as a leader? What do you do first? What sort of team do you put together in terms of internally, externally? Can you just tell us a little bit from your perspective where to start?
Sure, thank you. I have to say that the best way to start is to just start. you need to begin the process of collecting natural history data or identifying a source for natural history data almost as the very first step. And this often involves doing a literature search, making sure that you've identified experts in the field, and looked hard at any natural history that already exists that might be a guide for you. And then once you've decided that what already exists is not enough, then you got to put one foot in front of the other and plan a natural history study for yourself that's going to provide you with data by the time you're going to start your proof of concept trial in the disease area that you're interested in. So that could mean you need to start 2 years ahead of filing an IND, where you get the ball rolling. The team that you need has to be flexible, and they have to be rigid all at the same time. So they have to be rigid about data quality, but they have to be flexible about the overall plan, because there are going to be bumps in the road. There are going to be things that are just not possible to do or too difficult to do, and other things that are difficult to do but should be done anyway. And you sort of need to decide where you're going to put your effort and where you're going to back off and realize that there are some things that are just impossible. And I would say part of the planning process has to involve medical people, operational people, and statisticians. And if you don't put all of these people in the room at the same time so that they can all get on the same page, you're likely to make some mistakes. Back to you, Meganush.
I completely agree. And I have seen the examples of the successful process and not so successful process in this regard. We have a question from our audience. And the question is, was there any input provided from regulatory bodies in terms of the historical data being captured in Excel and not being 21 CFR Part 11 compliant? Mark, Nicole?
So this was not a concern in this example, because the main concern of the sponsor was to plan a clinical trial. And they did not have the intent of using this to support a single-arm trial so that this represented a historical control. If this was going to represent a historical control, it's, half of the data is way too sloppy to contribute. And that would be the data from the academic investigator. But what we're attempting to do is just increase the N. And because they're using a similar study schedule, similar outcomes, and similar exposure to the toxin, it can help us a lot in making sure that our statistical models work well. And it can help a lot because now we have some idea of what to expect from the biomarker, from the ClinRO, and from the PRO when we deploy a clinical trial. The sponsor needs to make a decision about whether their proof of concept study is going to be 4 weeks long or 12 weeks long. It looks from the data like the sample size has to be much larger if they're going to do a 4-week study than if they do a 12-week study. So there's a trade-off between the duration of observation and the sample size that they're going to need to detect their drug effect. And so they've learned a lot about how this is going to work, and we'll be able to make decisions about whether they want to do a shorter study with a lot of subjects or a longer study with fewer subjects.
Thank you, Mark. And I wanted to add a little bit to this. In my experience, and I know this is not applicable to this specific example, but in my experience from the interactions I've had with the regulators, especially the FDA, surrounding natural history studies, that there is an understanding and appreciation, especially in rare diseases, when your only source of information is academically, generated, is academic centers that are capturing this data, there is an understanding that they did not capture it with the same standards of clinical trials, and that there is still things that can be done afterwards to mitigate, afterwards to just ensure the quality of the data. And to the extent that there are SOPs in place in these institutions, and there are protocols in place for capturing these data and so forth, all of those documentation are very helpful to gather, put together, and provide in an informative package to the regulators in support of the data. Whether you're using it for hypothesis generation, especially if you're using it to validate a surrogate endpoint, let's say, or especially if you're using it as a historical control arm. That's been my experience. So regulators understand that it is what it is, right? But on the other hand, you as a sponsor do as much as you can to mitigate as much as you can, given the circumstances, to provide documentation and to be transparent. Thank you so much again for that question. We'll go to another question, and this one is specifically addressed to Chelsea. And Chelsea, you alluded to that the prospective natural history study, even though it uses a lot of the methods of clinical trials, but it's not the same thing as a randomized controlled trial or a clinical trial that involves a drug or biologic being used. And there are aspects that need to be considered by the sponsors and flexibilities and so forth. Can you give us a little bit more color on that? And I know that we will have a deep dive on the operational considerations in the second part of our 3-part webinar series. But can you please just provide for our audience a little bit of color around what you alluded to? Thank you.
Sure.
Some examples,
sure. Thank
perhaps,
you, Megan.
or one
yeah,
example.
Sure, of course.
Thank you.
Yeah, I'd love to. So I think I'll kind of piggyback off of what Mark was saying, that, with natural history trials, you have trade-offs. And it's also really important to have all the right stakeholders and the key stakeholders part of the conversation right from the beginning. Those things are the same as what you'd have in any clinical trial. I think the difference here is understanding the long-term plans for the data and making sure that you're setting yourself operationally up for that fit for purpose reason. So I'll give you some examples. One of the pitfalls that I talked about was losing patient and investigator interest. A strategy there would be involving patient advocacy groups and involving them really closely to your natural history studies. So using them as a tool to reach out and connect with those patients and understand the patient's lives, communicating directly to the patients if there's information that's needed in the registry, if you're losing interest in the registry, making sure you don't have loss to follow up patients happening, on your registry data, that's a strategy that you may not, tap into as much in a normal clinical trial, but it's a huge one for natural history studies, especially when you're talking about rare disease. When you're trying to find the patients to actually recruit them into the registry, making those alliances early is key. Another example that I might give is technology. So don't underestimate the use of technology in natural history and using it as a means for communication to that group that you're collecting data on. Technology can be expensive, but if you're looking for the natural history data to be, the long-term use of control arm for a study, it's probably well worth the investment to get data direct from the patient and have a means for having eSource so that, it'll also help your monitoring efforts operationally if you're getting that data direct from patients. And then the last one that I'll just mention quickly, I know you said one example, I'm giving you three, is your ICF, your IRB requirements. So the FDA has released wonderful guidance that has quite a bit of information that's helpful, but I would just encourage you to work with your CRO partner, your IRBs. If you're using a centralized IRB, make sure the language that's in your ICF is really speaking to how you plan to use that data and making sure that your patients are aware of how that data is going to be used. That'll keep you in line with the regulations. Thanks, Ganesh.
Thank you so much, Chelsea. This next question, I love this question, and I invite everyone to give their comments. I'll start off with at least some of it from my perspective. So the question is, do ProMetrika have any suggestions for making up time in gathering registry data, since this type of effort can take a long time? Pros and cons of doing a retrospective chart review versus prospective collection. So to me, this is a great question. In terms of my own experiences with this, and I'm trying to look back, I think a lot of times there is concurrent work that is ongoing by way of CMC, by way of other considerations that are going on. And while that is in progress, clinical teams focused their first efforts on natural history studies. Again, this I have seen a lot in the rare disease context. That's my answer to the part one of that question. And I will also ask the panel to give their comments on that.
I can jump in. I'll give you an example of where I saw a jump start in natural history and also sort of the response of subjects to compassionate use treatment. So I worked on a rare disease in which the most of the subjects in the world were in Taiwan, about half of them, and then the rest were scattered over the planet. And by the company involved a chart review of the hospital in Taiwan where most of these patients were taken care of. The company hired a Taiwanese CRO to do the chart review. And then there was a second step where the elements of the chart review had to be translated into English. Then those elements had to be transferred to a second CRO in the United States, where the TLFs were created and the data was edited, right? Because we had to sort of go through it and make sure everything was as it was supposed to be. And then we had to query back to the people who did the English translation, and then query all the way back to the Taiwanese paper record to make sure that the data element was correct. So we ended up having a team that was very specialized. The CMO for the sponsor company had incredible social skills. So he took care of speaking to the PI in Taiwan. The clinical operations person was extremely organized, and she took care of managing the Taiwanese CRO just to make sure that, you know, every field was filled in and we understood what was in every field, and every value was in the expected range or was queried. And then we had a CRO in the United States that was responsible for receiving the data, making sure that it was in the same shape that it was expected to be in, and then creating data displays so that we could understand what was going on with the subjects. So this was an incredibly intense effort, and we were working with about 21 subjects. But the amount of data that we had for each of the subjects was large, very large. And so the effort involved people just focusing on a piece, but at the same time participating in team meetings, making sure that we knew what was going on, and then half the team was up late at night talking to the Taiwanese because of the difference in time zones. So it's possible to do, but the real way to get a jump start is to have a data source already identified that you can mine, and then make sure that you put a big effort in mining that and making sure that it's cleaned up.
And from my perspective, this question of, thank you, Mark, by the way, the prospective and retrospective, I wanted to elaborate a little bit. Of course, if you're going to use the data as a historical control arm, as a synthetic arm, right, to compare to your single arm trial, if you have the luxury of having a prospective study, of course, that is preferable because of your visit structure can be set up accordingly to make it fit for purpose for that comparison. And, you know, the operational considerations, the quality of the data, you have much more control. And the disadvantage is that it could be a lot more expensive. It could be as expensive or maybe slightly less expensive than a clinical trial of the same size. So that's a consideration, of course. In terms of the retrospective, the challenges are basically fitting things that don't naturally fit together and trying to compare visit structures that are different. And of course, if they're data that come straight from an EHR, there's a lot of variability to the data. There's a lot of lack of structure that then you need to post hoc impose the structure on it. So it makes for a very fun and interesting statistical exercise, but of course, if it's possible to avoid that and have something that's prospective, that's preferable from my perspective. We have another question and a comment from one of our audience members that retrospective data can be a nightmare. For prospective natural history data, we need attention to the data collection as it evolves, or it may be as messy as retrospective data without operational controls. This is a comment from one of my dear colleagues on the sponsor side, and thank you so much for this comment. And I absolutely agree. I absolutely agree. Thank you. Going back to our case study, I know I'm excited to find out, Mark and Nicole. I know, Nicole, you alluded to some of the next steps in terms of the model building, but what are our goals? What are we doing with this modeling and what is next? And I don't know if you can share, given that this is an ongoing project and we have taken pains to make sure that the identity of the project is protected and so forth. But can you share a little bit what's next? Nicole, from your perspective, what is your next steps?
So from the modeling perspective, as we said, we're going to start with the sponsor data because that data is already clean and we have that available. So we're going to build some simpler models with that data because we only have 19 patients in that data set. So we can't obviously, build models with many parameters. We have to build very, very tight models. So we're going to start with that. The goal is to basically to build growth models and to see how both the PRO and the ClinRO scores evolve over time and how they can be predicted by predictors such as cumulative dose and baseline covariates. So we're going to be using PROC-mixed. We're going to be likely we're going to be including random intercept, random slopes in the models. And eventually, once we get the academic data, the hope is that, we'll run these models again that we've developed with the sponsored data. And hopefully we're going to see that they're consistent, that the model fits and the parameter estimates are consistent between the sponsor and the academic data. And then eventually, combine both the sponsor and the academic data, because that will give us the biggest, data pool from which we can build the most robust models with even more predictors potentially. But I'll let Mark speak to exactly what the objectives of the model building process is.
Sure. So we need to know whether there's a time course for the biomarker, for the ClinRO, and for the PRO. And whether that time course is simply an artifact of the cumulative dose of the toxin that we're giving, or whether the time course itself is important. In other words, which of the two variables is the dominant variable. And then we need to know sort of when there's a change in the biomarker, the ClinRO and the PRO. It could be completely linear, or it could be that it looks like a step function where there's a threshold effect and at low doses of the toxin, not much happens until you hit a threshold and then suddenly the ClinRO jumps up or the PRO jumps up or the biomarker jumps up. So we need to be aware that we're looking for those things. And until we start with some basic graphical analyses and then do some statistical modeling, we won't be ready to do additional graphical displays that'll be informative as well. So it'll kind of go back and forth between doing visual displays and doing modeling to make sure that the variables that we're displaying are the right ones. And that will lead us to understanding the data. And that's why I was saying it's important to know whether we're going to do a 4-week study or a 12-week study and what our primary outcome variable is going to be for proof of concept, and which one should we use that's going to give us the smallest sample size and the easiest chance of proving that we have a drug that can prevent this toxicity.
Wonderful. It's exciting, very exciting. And we're very happy to be a part of that journey forward. Thank you, Mark. So this brings our first session of our three-session webinar series to an end. I would like to thank all of you, our audience members, for attending today. We have a great turnout. It's exciting to see. We are honored, and we hope that you can join us again next time. Please watch out for emails from us with information, with follow-up information about this session today and future sessions. And as Chelsea had mentioned, we will include some pre-specified times that if you have questions, if you have interesting sort of experiences to share and you wanted to talk to any of us one-on-one, we will provide some times for that. Thank you again, everyone, and have a great rest of the day.